Hi, everyone, and welcome to episode 36 of Diabetics for Diabetics Radio. Today's episode is titled Harmine and Beta Cell Regeneration. We are going to talk about the plant compound making noise in diabetes research and what that means for you and I, those dealing with diabetes. If we haven't met yet, my name is Bowie. I've had type 1 diabetes for over 22 years. I am healing naturally and sharing information as I go. About 2 years ago, I took a deep dive into a topic, specifically iron overload. And in learning more about this alternative health type mineral balancy aspect of metabolic health, I learned so much about things that everyday people could be doing for their blood glucose, could be doing for their metabolic health, and it ballooned into this hope and this faith in the idea that there is something that we can do with diabetes to get to a point where we don't have diabetes anymore. That's where this podcast comes from. That's where my social media presence comes from. I am on TikTok as well. You see my handle there at Bowie_Madison. Don't hesitate to follow me to stay up to date on what it is we are doing in the diabetic realm. There are so many people out there doing the most to handle their diabetes on their own and get to a point where increasing insulin sensitivity, decreasing insulin resistance, improving metabolic health, and decreasing the amount of insulin they are using on a day-to-day basis. So, welcome if this is your first time. If not, welcome back. Let's go ahead and get started. And we are diving in without any lead up, okay? Right into a paper title immediately, okay? So we're going to start talking about harmine, how it was found, what it's done, and then we'll kind of like break it apart into pieces and talk about how it fits into the whole here. So this paper was, I believe, published in 2015, and it's titled "A High-Throughput Chemical Screen Reveals that Harmine-Mediated Inhibition of DYRK1A Increases Human Pancreatic Beta Cell Replication." Now, for those without biology and chemistry degrees or any experience reading scientific papers, it sounds like a lot. But all we're really interested in is this right here: increases human pancreatic beta cell replication. Okay. They've found something that leads to beta cells being able to replicate, which, as someone with type 1 diabetes, is what we've been told is the issue, right? We have these, whether it be the autoimmune system or vitamin and mineral imbalance that has caused our beta cell mass to decrease, thus decreasing our insulin. It's been unable to regrow to a state that keeps us alive, right? So we become and remain dependent on exogenous insulin. So beta cell replication, I like where this is headed, okay? So in this paper, they took a huge amount, like a huge database of different compounds, and they tested to see what effect they would have on growth factors in beta cells. And there were a handful that came back as being better than all the rest, right? And that's where harmine got introduced. That's the plant compound. It was very effective at inducing cell growth, and it inhibited a specific set of protein kinases that were tied to stopping cell proliferation. So this makes sense. Something that stops the inhibitor, right? If you stop what's stopping beta cell growth, beta cells grow. And that's kind of how they figured out how this was doing what it was doing. One of the things they discovered was it was not specific to beta cells, right? So the effects that this compound had on cells' ability to regenerate was not just beta cells. It was alpha cells. It was all different kinds of cells. And that sheds light on one of the issues of beta cell growth is we want something as people with diabetes that is going to target beta cells specifically without growing anything else. If you grow things that you don't want to grow, you are promoting imbalance. That's where things like tumors and cancer come in as well. You really don't want things to grow outside of the scope of where you actually are wanting to grow. But one important point here that I thought was unique and caught my attention was the fact that they tested this beta cell growth in a number of types of diabetes. And what I mean when I say types of diabetes is they tested a lot of different ways that diabetes can develop. The first is partial pancreatomy. So they had mice whose pancreas was partially, like, maybe we'll say cut in half. I don't know if it was actually cut in half, but a piece of it was taken off, right? And that compromised the mouse's ability to stabilize its blood sugar. They had the immunodeficiency model, which had a mouse with an immune system that was lacking a lot of the key white blood cells and leukocytes that are responsible for protecting beta cells. And then they also had the inflammation model, the inflammatory model, where a specific inflammation-inducing compound, streptozotocin, which we've talked about on this podcast before, is injected, and it has a targeted effect on destroying beta cells. Okay. A lot of the times in diabetes research, what you'll see is they'll kind of do like a cover, like, oh, how do you make diabetes in the lab? And a lot of the times people use streptozotocin because it's an easy, you inject the mouse, the beta cells die, you now have a mouse that is exhibiting the same symptoms of someone with diabetes. This one looked at it in a bunch of different ways to see how versatile the growth effects were of harmine, which is the plant compound, okay? So let's get introduced, right? Harmine was this plant compound that was showing all of these incredibly promising results in getting beta cells to grow. So harmine is a harmala alkaloid. Alkaloids are a very popular plant group in health and wellness because of their antioxidant qualities, their abilities to mitigate oxidative stress. It's a beta-carboline, which has to do with its shape. You have all of these carbon rings here. Again, associated a lot with being an antioxidant, managing oxidative stress being very good thing for health. If you were to Google beta-carboline and the types of things that harbor beta-carbolines, it's all like very healthy food, vegetables, fruits that you'd be familiar with. It's also a monoamine oxidase inhibitor, MOA, or MAO actually. And we'll talk more about this, but this has some societal and drug implications. We'll get into that in a little bit. But again, there's a lot going on with this harmine molecule. And I thought, interestingly, it was tested as a treatment for Parkinson's in the 1930s. So it has some very strong connections to neurological disorders. And so harmine was actually something that was used with success in managing Parkinson's disease back in the 1930s. So it's a plant compound, right? So, okay, what plants? And passion flower is one of them. Syrian rue is another one, which is a Mediterranean kind of like a desert shrub, desert bush. Aztec tobacco, Nicotiana rustica, I believe. Yeah, Nicotiana rustica is the scientific name for it. And then this is where that MAOI, the monoamine oxidase inhibitor and the psychedelics. It's also found in the vine of the gods, which is Banisteriopsis caapi, which you may or may not be familiar with, but it's actually a component of ayahuasca, which is the tribal medicine that is very popular in the Amazon, but really it's grown in popularity worldwide for its neurological, psychological, spiritual health benefits. And harmine is actually one of two psychoactive components in ayahuasca. It's sourced from a number of different sources. In the traditional ayahuasca brew, it's this vine of the gods, the Banisteriopsis caapi, that is usually where the harmine comes from. Harmine, the monoamine oxidase, actually synergizes with what we associate with being the most active component of ayahuasca, which is dimethyltryptamine, or DMT. That's kind of the, LSD rainbow light show drug that we associate with using ayahuasca. But actually, harmine is basically like the key in the ignition to allow for DMT to have its amazing effects. So just an interesting tidbit, and something we'll consider later on when we think about how, with all of these wonderful things that harmine can do, what might be some of the limitations when it comes to implementing it in the general public. So I mentioned before with the MAO pathway, one of the most critical components of harmine, one of its most potent targets, is the DYRK1A pathway. Okay, and DYRK stands for dual specificity tyrosine. Tyrosine is abbreviated with a K. That's where that K comes from. Tyrosine regulated kinase. Okay. I think I got that wrong. Dual specificity. Tyrosine is with a Y. Sorry. Tyrosine is with a Y. Regulated kinase, DYRK. And then there are a couple different subsets of the DYRK pathways. 1A, there's 1B, there's 1C, I believe, as well. But don't get lost in the sauce. Actually, when I read DYRK1A, I say in my head, DYRK. So, if I'm reading a paper that reads that out, I don't read out DYRK1 every single time. I just say DYRK, the DYRK pathway. So this particular pathway, the DYRK1 pathway, involves enzymes that correlate with cell proliferation and brain development. Okay, so again, ties to neurological disorders. We talked about Parkinson's before. We talked about the psychedelic and the spiritual component of ayahuasca. It has a lot of ties to disease. And when you think about dealing with cell proliferation, whether that be an overgrowth of cells, which we think of with tumors and cancer, or an undergrowth of cells like we think of with beta cells and diabetes, it makes sense that this would be influential in the health of cells. And sure enough, I've got a paper here titled "The Omnipresence of DYRK in Human Cells." So this just goes on to say that DYRK does so many things to not be important, right? And if there is any sort of dysregulation in DYRK activation or inactivation, you're going to feel it, right, in the body. So DYRK is a subclass of protein kinases. I think I've mentioned this before in podcasts previously, but protein kinases are like on-off switches in the body, right? These kinases are things that go around and turn proteins on when they're needed, turns proteins off when they're not. This is just one specific case of protein kinases, the DYRK. So DERCA has multiple biological functions, okay? Cellular signaling, mRNA splicing, chromatin transcription, DNA damage repair, cell survival, cell cycle control, neuronal development and functions, and synaptic plasticity. I mean, this is like an all-star protein kinase, okay? So cellular signaling, we're interested in insulin resistance. We're interested in beta cells knowing when to grow and when not to grow. mRNA splicing and chromatin transcription, DNA damage repair. This has everything to do with the creation of proteins. It has everything to do with the regeneration of proteins, healing damaged DNA in states of oxidative stress, which inherently ties into cell survival. So this is like such an important component of a healthy cell cycle. It's definitely something that we need to be paying attention to, and it's not surprising at all that targeting this is something that would benefit those with type 1. So harmine is seen as something that gets beta cells to start to proliferate, which is something that is not easily done, or so we're told. So harmine inhibits the DERCA pathway, right? So it's inhibiting the inhibitor. DERCA is something that stops beta cells from proliferating. By stopping what's stopping proliferation, you're actually promoting more beta cells. So in that initial study that I mentioned at the beginning of the podcast, in each of the different origin styles of diabetes, whether it be pancreas is physically damaged, whether it is immunologically damaged, or whether it is inflammatorily damaged, right? Either it's cut in half and you lose part of your pancreas, the immune system isn't able to protect it, or there's just so much oxidative stress going on in the pancreas that it ends up dying and the beta cells stop. Harmine and its effect on the DERCA pathway was able to induce growth in all three of these models. Of note, it was not specific to beta cells. It was able to grow beta cells, but it grew a lot of other things as well. And alpha cells was one of those things in the pancreas. And I've also mentioned this before, talking about how diabetes isn't just regrowing beta cells, it's restoring the balance of alpha and beta cells. So this is a little bit of an obstacle when it comes to actually treating type 1 diabetes, because it's growing beta cells isn't enough. It's a matter of also stopping the growth of alpha cells to allow for beta cells to kind of close the gap and restore balance between the two. Good news, though, last bullet there, no observed DNA damage or cell disruption. So this wasn't something that was like, if you use too much, the cell goes kaboom and it's worse. You're worse off for using it than you were not at all. So this is something that has therapeutic potential. So I mentioned it just briefly before. Diabetes is about the balance of cell types in the pancreas. It doesn't matter how much your beta cells grow if your alpha cells continue to grow too. Diabetes itself is a balance of alpha and beta, not just more beta, okay? So if you're diabetic, you have overactive alpha cells, you have underactive beta cells. Harmine on its own was shown to increase proliferation in both alpha and beta. So an increase in beta cells would only be part of that equation. How do we make a targeted increase in just beta cells without also increasing alpha cells, right? Where diabetics are alpha heavy, beta shy. We need to get those beta cells up without also increasing, right? It's the difference between the two that is causing diabetes. We need to restore the balance. So the targeted regrowth of beta cells would increase insulin capabilities without having to overcome the pre-existing and overactive alpha cells, right? We need more insulin, less glucagon. So what's something that we can temporarily use to inhibit alpha cell activity? What is something that we can do that's going to inhibit the liver's glucose output, which is constantly sending people with diabetes blood sugar up without having the ability to bring it back down with insulin? Think about that for a second. Boom. GLP-1 agonist, glucagon-like peptide 1. And sure enough, this was about 5 years after the initial paper that I showed at the beginning of the podcast. GLP-1 receptor agonists synergize with DERCA inhibitors to potentiate functional human beta cell regeneration. This is exactly what we were looking for, right? We needed targeted increases in beta cells without also increasing alpha cells. So what this paper found was that when you take the DERCA inhibitors like harmine, which are going to promote cell growth, non-specific cell growth, but it's able to kind of get the juices flowing and start increasing numbers. When you combine that with a GLP-1 agonist, which is something that's going to inhibit specifically alpha cells from proliferating and from activating, now you have this perfect combination that is going to get targeted beta cell growth. Okay, let that sink in for a second. When you use harmine, it is indiscriminately growing everything it sees in the pancreas, right? Beta cells, alpha cells, gamma cells, delta cells. We want it just beta cells. So we are taking this growth factor. At the same time, we are stopping the growth of alpha cells so that all of that growth potential is being delivered directly to the beta cells. And it worked in this paper, okay? So increased rates of beta cell growth. I'm just going to read this out from the results section of that paper, which I have titled here, and I'll have a link for it in the description. So harmine and GLP-1 synergistically increase human beta cell proliferation. We selected GLP-1 as a potential partner for a DERCA inhibitor due to its superior human beta cell targeting specificity compared to DERCA inhibitors. Okay, so GLP-1s have been shown to allow for beta cell regeneration before. Not that they specifically target beta cells and grow them, but they allow for their regeneration a little bit better because of their alpha cell silencing. Proliferative synergy is a class effect for DERCA inhibitors and GLP-1 agonists. So this is a really, really cool component of using these two compounds together. It's that you take each of them alone, and they're both good. When you combine them, their effects are magnetized. Okay, so the synergistic ability of harmine to drive beta cell proliferation in combination with GLP-1 was apparent for every DERCA inhibitor tested. Now you see there's 5 of them here: harmine, Indi, lustatine, 5-IT, and GNF4877. Those are all different kinds of DERCA inhibitors. And they combine them with a bunch of GLP-1s, and I'll mention them in just a little bit. But thus, proliferative synergy with GLP-1 is likely a class effect. So almost all DERCA inhibitors work synergistically with almost all GLP-1s. Okay. They also found that synergy was apparent for harmine in combination with all 5 of the GLP-1s that they used. Okay, this result indicates that mitogenic synergy, which means cell growth, like mitosis, mitogenic synergy with DERK inhibitors is a class effect for GLP-1R agonists in general. These are the GLP-1s they used. A lot of them you are probably familiar with. Exenatide, Victoza, Lixuma, Adlixin, and then the last one, which is the most popular, at least here in the US, Ozempic, which is a semaglutide, Rybelsus, Wegovy. These are all things that have been targeted for their ability to decrease liver output, decrease alpha cell activity in the pancreas. So pretty neat. And when you actually look at the statistics of growth, it's pretty impressive, right? Okay, so let's look at this graph. This is from the Akifi paper from 2020. So blue cell growth here, the blue, is just the DERK inhibitors alone. So just harmine and all the other 5 that they have, Indy, 5IT, GNF, I think it was 24877 or something like that. But a modest amount of growth for those blue. Now, look what happens when you add that GLP-1 agonist. You're almost doubling in every case the induction of growth, right? So Ki67 is a marker for beta cell for cell regeneration. So to see if a cell is growing, it would have Ki67 as a marker in it, right? So it's basically just a growth factor. If a cell doesn't have Ki67, that means it's not growing, it's not proliferating. So harmine alone increased a fair amount. By adding the GLP-1 agonist, everything almost threefold in this last example, but you're getting significantly more growth. And for percentages, right, if we're talking about how quickly it is for growth, standard growth for a beta cell is somewhere between 1 to 3% when it's stimulated with some sort of harmine. Like harmine was, I think, somewhere between 2 and 5% growth. When you added the GLP-1 agonist, you're getting anywhere from 5 to 8 to 20%. I think 20% was the highest they saw out of any of them. But that's like a huge improvement in the beta cell's ability to regenerate. So this is big news. So now, okay, cat's out of the bag. Beta cells can be regenerated with things that are relatively accessible to us, those with diabetes, right? So as with any research topic in diabetes, like stem cells, we've grown immune to any sort of positivity in the research. Why? It's slow. It's a very slow process. From getting funds, getting trials canceled and started and canceled again, bridging the gap between, oh, it looks really promising in animal models, why can't we get it to human trials, right? You kind of have to go through that noise. Harmine also has some social obstacles to overcome because of its association with ayahuasca and psychedelics. The spiritual realm is not at all welcome in the very kind of like clear-cut, cold, clinical aspects of modern medicine. Ayahuasca is something that, with a lot of different variables, that does not necessarily mesh with how modern medicine does business. Something to consider. And then for you and me, dealing with diabetes, wondering how we can implement this for ourselves, what do we need to be mindful of? One of the slides before, we mentioned that there weren't any disruptive effects of using harmine, right? Too much, too little. How do we know what the right dose is and what's too much? How long do you use it? That kind of thing. And so you have to experiment, and that's kind of what you see in those pictures here. So I was actually able to get my hands on Syrian rue, which, like I mentioned, is kind of like an everyday shrub in dry, sandy, arid conditions, a lot in the Mediterranean, some in the US Southwest. And what I did was I bought the seeds, I ground them up into a powder, and I stuffed them myself into capsules. And I started to kind of play around with combining harmine with natural GLP-1s. So going back to where does harmine come from? I chose Syrian rue because of its accessibility, because I could ground it up and put it into capsules. I have since tried passion flower tea, which is not all that uncommon. I've also used Aztec tobacco, Nicotiana rustica. It's not like your average cigarette tobacco. You're going to have to look a little bit harder. It's a little bit more of a specialty purchase. And then again, I can't condone any sort of illegal activity or anything like that, but people are experimenting with ayahuasca. I have heard anecdotal reports of people having success on these ayahuasca retreats. I haven't done it myself. I can't speak to it, but by the plant compounds and what's actually in them, I'm not ready to count them out. So you have your harmine, right? These are kind of your possibilities for getting doses of harmine. How do we get our natural doses of GLP-1s without signing up for all of those extra pharmaceutical exenatide, Ozempic stuff? Because that stuff is not to be relied on. Nature is rife with GLP-1s. You'd be surprised. This is just one paper documenting a lot of things that you can use that have a similar effect to GLP-1s. Berberine is one I'm most familiar with and have used the most. Resveratrol, which is a potent antioxidant. Curcumin, which is the active component found in turmeric. Green and black tea. Gardenia. Cinnamon, Ceylon cinnamon specifically, is usually what's recommended for people with diabetes, and soybeans as well. Okay. For those soybeans, you are obviously going to avoid genetically modified, processed beans. You want to stick to organic if you can find them. So I have been playing around for a good while now, probably over a year and a half, on combining these two, right? The DERCA inhibitors and the GLP-1 agonists. I've done the passion flower tea. I've done the ground Syrian rue in capsules. I actually have them right here. And then Aztec tobacco. I actually did buy Aztec tobacco as well. You can use it in a tea. You can roll it and smoke it in a cigarette. Any way. In higher doses, you're going to kind of like feel the effects of the monoamine oxidase. But again, you have to proceed with caution. You have to proceed with what you feel comfortable with. Of course, none of this is medical advice. Proceed at your own risk. GLP-1 agonists: berberine, Ceylon cinnamon, green, black tea, turmeric, and black pepper. These are things that I am combining with the DERCA inhibitors to see if I can get some beta cell activity. How I would be measuring beta cell activity: what's my insulin usage like? Am I needing as much? And here's what I saw, and I lost my mind when I first started with these Syrian rue capsules about a year and a half ago now. Within the first 4 weeks, huge decreases in my basal rates. Big time. I was on the pump at the time, and I was needing significantly less insulin, to the point where I was like, oh, like it was happening too fast for me. I was like, what is going on? That decrease plateaued after 1 to 2 months. And when you look, there are a couple more papers too that showcase that that's expected. If you use harmine too much, your body can adjust to that, and it kind of loses its effect. So I kind of have since been cycling it on and off as I've gone on. But I've continued implementing other sources of harmine: passion flower, tea, Aztec tobacco, and all mixed in with some form of GLP-1 agonist, whether that be berberine, quercetin, resveratrol, curcumin. These are all documented GLP-1 agonists of a natural origin. Now, why did it plateau? Why didn't it finish the job, go all the way? Why didn't I get totally off insulin? I didn't at the time account for any of the autoimmunity factors. I was offloading iron. I was rebuilding my mineral status to mitigate kind of the residual oxidative stress that I had going on at my day-to-day pace. But there may still have been something going on that was killing off some of the beta cells that I was regrowing. So I may have been taking 2 steps forward and 2 steps back, 3 steps forward, 2 steps back, 2 steps back, and then 1 step forward, right, and kind of breaking even at the end of the day. So beta cells may have been regenerating, but if my immune system wasn't calibrated, those newly grown cells would still be targeted. Okay. So now moving forward, we want to continue to harness the harmine and GLP-1's ability to boost beta cell regeneration. But now, how can we targetedly mitigate our autoimmunity and our immune system's ability to trust the new beta cells that are here and allow for them to flourish? Okay, and I've just gotten excited about the thymus gland and its ability to help reprogram autoimmunity. So I'm going to be experimenting in modulating the thymus gland to allow for what new beta cells I'm getting with the harmine and the GLP-1s to actually stay there and grow. Okay, so that's where I'm at with that. Harmine is a very, very interesting compound. And if you do a simple Google search of harmine and diabetes, you're going to get a lot of great resources and great papers talking about the effects of harmine on the diabetic model. All right. So that's it for episode 36. I can't thank you enough for being here. If you're not already subscribed, I recommend that you do. All of these incredible resources, not just for type 1s, but also type 2s, and things that you can do to mitigate inflammation, boost insulin sensitivity, decrease insulin resistance, promote proper insulin signaling in the body, and ultimately reshape our immune system to allow for all of this to flourish and allow for us to return to our natural state. Okay, so research paper links in the description, as well as a couple of resources that I have. I've got my ebook, I've got supplement links, I've got a nutrition guide, all there for the taking. Okay, so episode 36 of Diabetics for Diabetics Radio, harmine and beta cell regeneration. I am your host, Bowie. Thank you so much for being here. Enjoy the rest of your day.